please leave them in the chat throughout the presentation. I'll make sure we get to them later. And for those here with us in person, please hold your questions until the end. So to begin, in 2022, Katelyn graduated from Defiance College with a bachelor's degree in environmental science and started working as the Freshwater Research Technician for Defiance Research Alliance. Her days consist of conducting research in the field, collecting water samples with the water treatment plant, and in the laboratory processing samples and analyzing data. She has presented her research at several research conferences, such as the North American Diatom Symposium, the Lake Erie and Aquatic Research Network Conference, the Ohio Academy of Science, and the McMaster Symposium. Today, Katelyn will share her work and research on diatoms from the past 2 years as the Freshwater Research Technician. And with that, I will pass it over to today's presenter, Kat Smith. Thank you.
Hello, everyone. I'm going to share my screen, and then we'll get started with the presentation. We click one or...
Just go to click on the green.
Okay.
Yep.
Okay.
Your screen as well.
So is this my screen? So I can get it
Your
to...
split, so I can see you.
Okay. Oh, okay. Okay, so as Dana said, I'm going to talk about diatoms a little bit. And just to get a gauge of what I'm working with, we're going to do a little bit of an introduction of what diatoms are. I know a few of you have heard me talk about them before, but we're going to get started with some of the basics. As Dana said, my introduction, I graduated from DC. I'm the Freshwater Research Technician here at the college, and I also make time to help coach cross-country and track at Fairview. Okay, now we're getting into the diatoms. So they are a type of algae. They're very, very small, so you can only see them with a microscope unless there is like a large bloom of them and they become big clumps of golden brown algae. This picture here is what diatoms look like when they are alive. So they have chlorophyll in them. They are the start of the food chain, so they're important to the ecosystem. Their shells are made out of silica, so when I look at diatoms, I just see their shell. I don't see the green chloroplast in them, because when I process them, we kill the chlorophyll so we can see all the details on the diatoms clearly, which makes it easy to identify them. So diatoms are one of 12 types of algae. Usually you hear when there's an algae bloom, you're hearing about cyanobacteria. They're bright green, a little bluish color, and that's what we hear about when there's a large bloom, especially in Lake Erie. A true diatom bloom is golden brown, and you don't see those happen around here too often. There are different types of diatoms, and algae, but so when I look at diatoms, I look at plankton, which are floating algae, so diatoms that live in the water, flowing with the water. If it's a river or if it's a pond, they're floating in the water. And then benthic diatoms are attached to rocks or sediment or sticks that may be in the river or water that we're studying. So diatoms can be found in freshwater, but also they can be found in salt water. They can even be found in soils and caves and sometimes snow. So they're everywhere. And then diatoms do reproduce. So they can reproduce sexually or asexually. So diatoms are like the opposite of people. a young diatom is big, and every time it reproduces, its daughter diatom is smaller. So diatoms just keep getting smaller and smaller as they reproduce. So how do we connect diatoms to water quality? They are biological indicators, so they can tell us about the environment that they live in, depending on whether the pH is high or low, if it's salt water, depending on what nutrients flow into that water. Like around here, we have a lot of tributaries and streams that are running along agricultural fields, so we can tell different nutrient concentrations by looking at the diatoms that live there. The Maumee River has a lot of suspended sediment in it. That's why it's kind of brown and murky, because there's just a lot of sediment flowing in it. And you can, so different diatoms like that. So you can use all this information and diatom information to connect water quality. For example, some diatoms prefer a higher DO or a lower DO. Sorelia, I'll point that out later, is a type of diatom that prefers a high DO, which is dissolved oxygen. And orthophos is something that we talk about around here, or orthophosphate, and Fragillaria and Navicula are two diatoms that like that high concentration of nutrient. So I'll point those out later as well. So why care about diatoms? Why study them? So the background of this slide is an actual picture from a microscope slide that I took. So these are diatoms that I've found in the Maumee River tributaries that I study. So those big guys that look like crescent rolls, those are epithemia. Some smaller ones off to the side that look kind of like an eye with a slit in the middle of them, that's navicula. I see a lot of those. And then in the top right corner, there's one that just has a bunch of dots on it that kind of looks like a pea, kind of like the vegetable. That is Nitzschia. So why study diatoms? Like I said, they are the first in the food chain. So if they're not there, starting at the bottom and having little bugs and insects eat them and so on and so on, we aren't here to eat those fish that can be traced all the way back to diatoms. And I touched on it a little bit, that they are biological indicators. They tell about the environment that they live in. Concerning the area that we live in, we haven't studied diatoms in this area a lot in the past, and so getting a good set of base research on diatoms is good to have, so you can refer back to it. We also compare diatom data with chemical data, which I will explain a little bit in my poster research that I have. I have some graphs. So it's good to do a comparison, make sure everything's lining up, there's nothing out of whack. They can be impacted by humans, so we can impact them through eutrophication, which is nutrient overload in bodies of water. So the less we impact water quality, the less we impact diatoms, and vice versa. The more nutrients there are in a body of water, the potential for more diatoms, but those diatoms wouldn't be as biodiverse. So there would only be like a few species that really like that nutrient, so then they would overtake that bit, and we would rather have like a wide variety all living in a body of water. Okay, so some of the research I've done, as Dana mentioned, the McMaster Symposium I've presented at, same with OAS, Ohio Academy of Science. When I was a senior in college / my first year working as a freshwater research technician, I presented at the North American Diatom Symposium, which was full of other diatomists with lots of knowledge. It was cool to learn from them. And Dr. Brown presented a little bit about diatoms at IAGLR, which is the International Association for Great Lakes Research. And then this coming year, with the 2024 data that I'm working on right now, we plan to present at many of the same research conferences, as well as the Defiance Research Alliance workshop that we are planning for the spring to do a big overview of what Defiance Research Alliance is all about. I'll briefly go over my senior capstone project. I studied diatoms. This is how I got started with diatoms, and I did it on my pond at home. So we were able to find that copper sulfate, which is a treatment for ponds, does affect the algae, which diatoms are algae, and we treat our pond every so often because it is an aesthetic pond for us. We swim in it, and we don't want to be swimming with the algae, as nice as it is to study. We don't really want to swim with it. So I found that when we treated the pond, the algae levels were really low, so that means that that treatment is working for us. And it was cool to do a start out project of diatoms, which led me into all the research I've been doing with Defiance Alliance. So this is 2022 data. So this is the first year we really dove into the diatom research and got started. I'm gonna just talk a little bit about it on this, and then I have a couple graphs to go in depth so you can see better. These pictures of diatoms are some of the common ones that I found during this year and this first year of data. Since it was our first year, we just got a baseline, and we basically found that there were two zones with the diatoms. So as the seasons changed, so midsummer, spring, and then late summer and fall, there are two separate zones of data. And we also did some research with Hallie, and she focused on some flow and turbidity of and correlation with diatoms. So I think I'll get to the chart after this slide, but this is the area that I study, that we have worked on. So the Maumee, this is Fort Wayne. Maumee River runs through. Here's Defiance, and it flows north to Lake Erie here, and all the tributaries that we sample are in this area essentially. So for reference, this is the Upper Maumee watershed. A little bit deeper reference of all of the extra tributaries of Indiana, because it starts over there and then comes into Ohio. And so up close, this is what one of the graphs would look like. So this is the Bend site. If you know the Bend Road, we just stand on that bridge and drop a bottle down and get a sample. And so half of this graph, the left half, is diatom species, and then the right half is chemical data, which we get from the water treatment plant and the wastewater plant. I go out and sample with them on Tuesdays during our sampling season. This sampling season ran from about March to November, and you can kind of correlate some peaks with each other if you look at some diatom species here. Cyclotella, that's a round species, and it is a plankton species, so it likes to flow in the water column. You can see here early spring, summer, it did really good. There's probably a lot of water in the tributaries during this time. It also probably liked the temperatures better. You can kind of trace some of these spikes over to the chemical data, and that's how I do a lot of my data analysis, is comparing across to the chemical data, see if it can relate to anything that I'm seeing, and make inferences from there, do some article reading, see if other people have found similarly. And then if we keep over here by Cyclotella, you can tell in about mid-August-ish, it decides that it doesn't like the temperature, it maybe doesn't like the flow, maybe the rain during that time was low. There could be many factors during that time, but it drops off. And then these other species over here become more prominent. They like less water. They are benthic species, so they do prefer to attach to something and grow. And yeah, so this is an example of a graph that I look at. This is a little bit of Hallie's data from that year. She focused on flow rates and turbidity, and we compared, this is at the Cecil site, and so she found that ratios of benthon and plankton and benthon ratios were correlated with flow and turbidity, which we think might be interesting to look at this year as well. As we looked at the 2023 data this last year, we noticed some changes that we thought might be correlated with flow. So she did a lot of that data. This is 2023 data. We compared graphs, and these don't have the nice chemical data along with them, but this is what I presented this last year. We found similar findings. There's 2 zones from mid-spring to summer to late summer and fall with temperature change. We also saw different things. I'm gonna go to the next slide because I think I have another graph. Yes, okay, so this is Zuber, and this tributary is a man-made creek. It sometimes dries up. It sometimes gets totally cut off from the Maumee River. This year I actually went and sampled. The last 2 years I haven't. They would bring the samples to me and I would process them. But this year I was able to go out, take the samples, see the sites, which made a big difference. I know where the stuff's coming from. I got to see it every week. So that was really good. And this one gets pretty weird through the summer because it doesn't have a strong flow, and it eventually just gets cut off from the Maumee River, and it becomes like a pond of water basically that collects. This year actually it didn't dry up, but there's been a couple years where I just wouldn't have a Zuber sample, so I just didn't have that data. But you can see here the different diatom assemblages. Nichia and Navicula are prominent here. They're prominent everywhere else as well. I see those a lot on slides. So this is from last year, and we're, this last year. So, and then this next season, or the season that we've already sampled that I am analyzing right now and looking at, I'm thinking there's a possibility we could see a correlation with the drought that we've had this last fall. So it'd be interesting to see if anything connects to that, because the flow of the river got less and less as the sampling went on. Like there were some days like we weren't sure if we would get a sample from a river or not because it was all the way down at the bottom basically, and we could see the bottom of the rivers clearer and clearer every time. So it'll be interesting to look at that data for this season. This diatom on this slide is Eurosigma. It's probably one of my favorite diatoms. He's nice and big, and he's got a nice curve in him, so I think he's pretty cool. I develop favorites and not favorites because I look at them a lot. But this website is very helpful to me. If you ever want to learn more about diatoms, diatoms.org. They do a lot of cool things, and I met the people that run this website when I went to the North American Diatom Symposium. And if I ever am unsure of a species, I'll go to this page, look up what I think I'm looking at, make some comparisons, and there are thousands of diatom species, so there's more than you could ever know. And I only get to see freshwater ones, and there are saltwater ones, so those are all here. I think that's all I have. This diatom is Diatoma. He's cool. Sometimes he's really big, but sometimes he's really small. So I think that's all I have.
Questions?
Okay.
You can encourage me.
Hi, I've got a question.
Okay.
So it seems like your senior research project focused on your private pond, and your postgraduate work has focused on tributaries and the Maumee River itself, that watershed area. Were there inferences that you could draw from your initial research of your pond that correlated or did not correlate well with what you found then in the rest of the watershed?
A little bit. I would say I didn't directly compare any of the data, but I would say sometimes, like, while I was studying the pond at home, we were filling it up. So for a little bit of time, there was a hose that would run into the pond and that would create ripples like constantly. And so there was a little bit where I could tell like when the water was moving more versus still. So that connects a little bit to the rivers, how they're pretty much always flowing, but sometimes they can be still. So I think a little bit their connections can be made by having directly side by side looked at that data before. But the species were pretty much all the same between the two, even though I'm pretty far. I'm in the upper Maumee watershed, like our pond is, but it's pretty far from like any big tributary. Oh,
Yeah. Oh, there you go.
okay. Doug King, he said that Eurosigma is one of his favorites too. He said, my focus is mostly on diatoms, but have I ever sampled for benthic blue-green algae? I have not specifically. He said that some taxa like Phomoridium are known toxic producers and have mats that have caused pet deaths before. So that must be a thick algae. I have not looked at that before.
Good suggestion.
Yeah. You're welcome. Do you want me to ask or say your Jen English said, have you or has anyone in Defiance studied diatoms in the municipal reservoir? Yes. Jade, I don't know her last name, studied them through Dr. Brown. She was a student Dr. Brown had at Nebraska, I think, and she got some samples from the Defiance Reservoir core. So instead of water samples, they took a core sample. So you just send a tube down into the water column, into that sediment at the bottom, suction it, and you get like a tube of mud basically, and you can take layers and look at the sediments because the diatoms are in those sediments still. If they die, that's where they settle to. So I've actually looked at the pictures from those diatoms because I took pictures for her because we have a nice camera here on our microscope, and then she studied them. So I don't know how that research turned out, but yes, and I guess potentially they could be studied more since we have the pictures and she probably did the counts. And I think the summer research students from this year took some samples from it as well. But I don't know if they got into that research or not, or just were able to like practice sampling and get the research experience in, because that's something that the high school students did, I believe. Okay, I'll do.
Dan's is broader. Mine's related. First one.
Could I
Okay.
ask a technical question before you get to the big picture? Could you go back to one of your, what do you call those graphs? The cool area under the curved graphs?
Oh, I like on this
Yeah.
graph, underneath of it, like the percentages.
Just, yeah, what you call the kind of graphing. Anyhow,
Oh, there is a name.
like a cluster. Anyhow.
It is,
So-
yeah, it's like that.
Okay, so how did you So we're gonna need the right axis. I'm so sorry.
It's okay.
So to set up your two zones, what parameter did you fix your phylogeny on, your relatedness? Or I guess it was a population study, wasn't it?
Yeah, we run statistics through R, and
Yeah,
Dr.
good.
Brown would put all the code in, and then we would compare, and this is what it would spit out at us.
So,
There's Dr. Brown answer.
okay. Hi, Sabrina. I'm Julie Good. How are you?
Good.
This
Can you hear me?
Yeah, well, I'm here just to make Kat's life miserable, right? That's my job.
Okay. It's a constrained cluster analysis using the diatom count percentage data.
So, Kat, my question is that gray bar. So in this graph, the gray bar starts at like the middle of June.
Mhm.
But on the earlier data from 2022, it starts at August 2nd or 9th. So you were using all 10 parameters to dictate who clustered together? Is that what gave you your two zones?
I think so. I'm not 100% sure. I think so.
Well, and my underlying, my original question back when I first saw the data was, if you clustered it by like nitrogen ammonia level versus orthophosphate versus pH, would you see that zone change, or do they always sort together? Do your benthics and your planktonics always sort out that way?
I'm not for sure. I would say they always sort out that way.
Okay.
But
Yeah.
The data analysis is sometimes where my knowledge is still growing. But yeah.
Maybe that's the answer to Dan's question. So viewers might need to answer my question about the conversation.
Yeah, there was a correlation with it.
There was. Is that what we use to treat the algae in the reservoir?
Oh, I don't know.
One of the things that we use.
Yeah.
I just wondered if that.
Okay.
Dan had a broader question.
What are the next steps in your research with the Defiance Research Alliance? Right now, like currently, I'm counting diatoms. So when I count a slide, I count to 300 and I log them in, and then we do the data analysis to make these charts here. And I'll finish that up. We'll do the analysis in February, hopefully, so we can get it ready for presentations and conferences. This is the third year of the grant, so my sister is working and she's doing a potential research project as well. I don't know if she's narrowed it down or not. I think she's talked about doing some like native plant stuff. And we also recently went out to the BMP Best Management project that we have started with the Defiance Research Alliance, which is a self-forming stream / two-stage ditch project that we're gonna hopefully just take some pictures on and get information through that to see the progress of it, to see how it starts to form. And also there's some native species that were planted out there along that. Yeah, that's what I would say.
Are you going to continue to study diatoms? Will you continue to like expand and build on?
Probably. This year I hope to get the flow data specifically, since we measure the flow every time we would go out and sample, so there's a measurement. So maybe seeing how that graph changes with the different diatom species, especially as the drought happened this year, I think that would be cool to look at.
Did you say the word diatomists?
Diatomists.
Diatomists, that's the name for a specialized
Person that
researcher,
studies
a biologist.
diatoms. Yeah.
Okay, thanks.
How did you gain interest in diatoms to begin with?
primarily through Dr. Brown. I started out doing a summer research project when the summer undergraduate research program started here at DC. So I applied for that, did work over the summer of 21, and we studied diatoms because that's what Dr. Brown is really good at studying, or one of the things that she's good at studying. So we studied diatoms and tributaries around here, Upper Maumee Watershed stuff. And we also did macroinvertebrates, so the tiny bugs that live in the water and the leaves and stuff. And then I just followed it up that next year by doing a project on diatoms with my pond. And it was just an easy, not like easy because I didn't want to try something new, but easy path for me to take to increase my knowledge, and something that I started to learn about in the summer. And it's just kept increasing, kept being able to study it. And not a lot of people know what a diatom is, so it's cool to talk about them, and because they're everywhere, you just don't know it if you don't know it. Like, I didn't know what a diatom was before I did summer research that summer, so.
So you talked about the human impact
Mhm.
on diatoms. Like, what are the implications? Like, for example, some of your slides showed a lot of diatoms, and then it would show them like going away. Like, where did they go? Did they move on? Do we eliminate them? Do we?
So yeah, human impact on the diatoms. They don't really go away like in the graphs like this. They don't really go away. They just don't. They maybe aren't reproducing as much, or the conditions for them aren't what they need them to be. So they could potentially like die out and settle like after they've lived and gotten stuff from their like gotten the sunlight, chlorophyll, done all that. They could have settled to the sediment and just be done for that year basically, or that season.
What's the part of their life cycle that overwinters?
Say that
So
again.
do they have to be reintroduced, or do they insist and overwinter in that?
Yeah.
In that way?
They overwinter.
So it's kind of like a migratory plots. It's where they flow.
Kind of where they flow. You can find them in ice technically still. I haven't done any research in that regard, like actually look at the ice to see if there's diatoms on it, and like if they are alive or dead, like whether they have chlorophyll in them or not. I haven't looked into that, but Dr. Brown would always say we should get some ice and see if there's any in there. But
Good next step.
Yeah. If there are no more questions, we're gonna do a concluding remark. Thanks. Thanks all. Thank you. Dana's gonna close us out.
Thank you, Kat.
Okay, so first, thank you all for coming today. As a reminder, Julie Good from Allen County Soil and Water Conservation District will be presenting her edition of the speaker series on February 5th, 2025 at noon. Her presentation will discuss water sampling in the Maumee Watershed, talking about nutrients and sediments specifically. And thanks again for attending. There will be a recording of the presentation available by request, as well as any of those charts, graphs, or posters that Kat showed us. You can get those by responding to the email that you received the Zoom link invitation in. That's all. So thank you guys for coming.
Thank you.